Hello and good morning, good afternoon, good evening from wherever you are dialing in from. Thank you so much for being here today for our second day of our second annual Autoimmune Community Summit. I'm Lily Stairs and I am the board chair of the Autoimmune Association. I also have the honor of being the architect of our Autoimmune Community Summit. I am here and I do this work because I am a 3-time autoimmune patient. I'm the triple threat of Crohn's disease, psoriatic arthritis, and psoriasis. I fight for this community, and the Autoimmune Association fights for this community because we believe that there is a brighter future out there for all of us. And that's what you're going to hear about today on this brilliant panel. You're going to hear all about the innovative research and treatment development that is happening for our community. I was chatting with our CEO, Molly Murray, just a few days ago, and she said, God, these people are going to put us out of business. And we would love nothing more than that to happen. So without further ado, it is my distinct pleasure to introduce Steve Hoffman. He is the Associate Vice President of Research Partnerships at the Foundation for the National Institutes of Health. And he is here to talk with us today about the AIM-AMP program, which is focused on autoimmune and autoinflammatory diseases. So, Steve, please take it away.
Thank you, Lily, and thank you to Molly and the association for allowing us to talk a little bit about some of the big healthcare partnerships that we're doing today. So let's jump right into the slides because I know our time is limited. Move on to the next slide. So I want to make sure you folks understand what the FNIH is. We are not the NIH. We are not the federal government. We're actually a 501c3 that was congressionally mandated and developed to be, for the most part, a conduit to bring private companies and organizations and both industry, advocacy, and other venues to be able to co-support the mission of the NIH. And we do that through multiple mechanisms. Some of what I'm going to talk about today in these large research partnerships, but we also do things like support fellowships and endowments, build buildings on the NIH campus. So really a broad variety of mechanisms and ways that we bring the public and the private sector together to support biohealthcare and move the missions of the NIH and the missions of many of these organizations forward. Next slide. You may have heard of us on a couple different things. I'm going to talk about, as Lily noted, on the Accelerated Medicines Partnership and what that is and insights into our autoimmune efforts. I'm also going to talk about one of our other partnerships, the Biomarker Consortium, very briefly. You may know us, though, and it's easy to point to some of these large efforts across various types of diseases. We do a lot of work across many therapeutic areas, and this includes the active work, and that is really the warp speed efforts on the vaccine side. The treatment therapies that were done in combination to attack the recent pandemic, and even some of the variant analysis. So we've been involved there, as well as, again, as you can see from some of these other programs that are very impactful and very progressive partnerships that we're involved in. Next slide. So what is AMP? AMP really is about identifying novel targets, novel technologies to develop new therapies. And we've done this across the broad different therapeutic areas. Some of the original projects, this stems back to about 2012, 2013 when this all kicked off. And we've been able to raise and run these programs of greater than almost $1 billion now, which is just tremendous across Alzheimer's, type 2 diabetes, RA lupus, and the autoimmune conditions I'm going to talk today. But the success of these early projects then begat several other areas where we went into some of the neurodegenerative diseases and Parkinson's and schizophrenia and the neuropsychiatric, but also expanded into 2.0 efforts and phase 2 efforts of these programs to really make a difference as we've advanced technologies, advanced understanding of the diseases, and really found novel targets, novel pathways for new drugs that are not the ones that you see on the television ads today, but really are attacking things at the source, in our case, of autoimmunity. Next slide. If I jump into this sector, which is so important, and I since the whole time I've been at the FNIH for 10 years, I've been overseeing our inflammation and immunity portfolio of projects. So I've been very engaged in the autoimmunity community. And, some of the same questions still pester us today. We still don't understand some of the origins of the autoimmunity that's there. There's a lot of non-response to the various drugs that are out there. And we know there's a lot of comorbidity and a lot of complications that occur. So it is possible that as the technology has advanced, as our ability to dig deeper into the tissues, into the cell types that are driving the disease, could we find somewhere there's I guess a cross-pollination at the origins of that? Are there similarities? Why are there differences? Why is my drug different than this other disease drug? These are the things we're now trying to figure out, and we can look at the single cell level to figure out what these differences are. So those same questions that we're been tasking in the autoimmune and inflammatory areas are the same that still have pestered us and challenged us since the beginning. Next slide, please. So what we have done in the first AMP project, which was called AMP RA Lupus, is what we've like to call disease deconstruction. And so here we've gone into, on the RA side, looking at the joint, the synovial fluid and tissue that's in the joint, which is really the large source of the inflammatory response that's occurring in that disease. And then obviously lupus, a lot of the lupus nephritis and early parts of the lupus disease is certainly at the origins in the kidney. And so we therefore went after that tissue and tried to develop very advanced technologies to be able to look at both the tissue, matched blood samples from those patients that were involved in those programs, but also looked at the cell types. And we applied these new technologies, these single cell technologies, to look at the RNA, to look at the DNA, to look at the mediators and expression on the different cell populations that look to be causing these diseases, or for those that when people progress to different states of the disease, they progressed and had a more worsening condition. Could we identify what the different cell types are? What cells are coming in and infiltrating into and interacting in the actual site of the disease at its origins? And we hope and we believe that will identify these new targets, these new pathways that we could make druggable targets and really get more personalized, because we all know your disease is not your neighbor's disease, is not your friend's disease. They are very different in the way they're expression and the way they're causing these issues. This took a lot of work and consortium, and this is really where this approach of public-private partnership is really beneficial, to develop these standard operating procedures, to advance these technologies. It's different how you treat the cells in the kidney to get them ready to do these advanced technologies and techniques than it may be in the synovial joint or in the blood. And those minute differences are really important. And then when you start talking about bringing all these different types of data together, think about what's in the tissue, what's in the blood, all the different types of results that come out of this. Even the bioinformatics and the way we analyze all that data and cluster it together, that advancement needed to happen too. There's a lot of wealth of data, but you can get a lot of wealth of data and be paralyzed by that. And so getting that expertise together, both from the private sector and the industry folks that are doing this, the NIH, the FDA, and as well as our advocacy partners to really understand what the patient quality of life needs to be and where they want to focus. We wanted to make sure all that aligned in these large partnerships. Next slide. So when I look at this, a little complex figure, but on the left side is kind of what we've done in this disease deconstruction. We've done this in RA lupus, and now in this new AIM, autoimmunity and immune-mediated diseases program, which is kind of like the 2.0, we've started to add additional autoimmune diseases, psoriatic arthritis, psoriasis, Sjogren's disease. And we're doing these same techniques and technologies, looking at the RNA, the DNA, looking at the cells, looking at the pictures of the histology and advanced histology to really understand what are the different pathways, cell types that are involved. And now we're moving to the right side of this document in this program. Also, we're looking at these things in a 3D, right? It's important now that we might know what's going on in the tissue. Can we look kind of in the 3D space and see how they interact? And there's other things that we call metabolomics and lipidomics and different types of antibody staining that can be done to really look as how we call it the interactome. And this is how the different cell types are interacting. And again, those mediators that are causing inflammation, that are causing progression of your disease, they're important druggable targets, and really identifying which ones may be the same or different in RA versus lupus versus psoriatic versus the many other autoimmune conditions that challenge us. is really the core of this effort and what we're doing. Next slide. So this just shows the groups that are at the table, and I want to really enforce here that you have 7 NIH or 7 NIH institutes total and centers that are involved in this. The five main groups are here that are providing support. And the way the AMP works is we have NIH strong support that's basically half of the funding for this, and then we go out and we fundraise and get the other private sector partners here, which is a strong combination of both industry and the patient advocacy sector. Very pleased that the Autoimmune Association has recently came on board to be part of this effort. So very much engaged and look forward to working with them. But that patient voice and that patient advocacy sector and the educating of the patient of what we're doing, translating science to the patients, is critically important. Next slide. So we're really beginning to mature this plan. I talked a little bit about how this works, and I talk about these core diseases that we're focusing on in the beginning, and we're looking now at the same type of similarities. I talked about in RA about looking at synovial tissues in the joint, but we're also doing the same in skin, because as we know, a lot of, and certainly in psoriasis, certainly in Sjogren's, certainly in lupus, there are also skin similarities and differences across those diseases, and as well as pulling in Sjogren's, we can also look at glandular tissue that's involved in the dry mouth and those types of things that are problematic in these diseases. So these programs are to look at deep dives into the individual diseases, but they're also looking at these cross diseases where the tissue and the blood similarities could provide some really interesting results. And then obviously, then we want to look across all autoimmunity and see if there's drugs that are out there that could work across the different ones. But then when they don't work, what is the reason why they don't work, and what are the other potential pathways we can target and provide better personalized medicine for you and all patients. Next slide. I don't want to jump into some of this, a little text heavy, but some of the big questions. We want to understand the early disease, not just the non-responders, but can we get to the point that we understand where the early modifications of the disease, when they're right at the beginning of it, can we attack there? Maybe we even take things for people that are high risk and start them on therapies in an earlier window of time, so you don't progress to these other systemic inflammatory responses that go on. Such great heterogeneity in the types of disease. As I made the note about, your disease doesn't look like your neighbor's disease. We need to understand what are the cell types involved, what is the differences individually between people, so we can personalize that medicine, personalize that treatment therapy for what we're doing. And again, by doing this and integrating all these different technologies and deep dives into at the single cellular level in the tissue and the blood and the skin and the glands, combining that analysis and these technologies, very, very powerful tool. And simply 5-10 years ago, we did not have the technology to do that. Now we do, and now we need to interrogate these data and really advance the science. Next slide. So we talk about where those unmet needs are. You know them, you live them, and the identification of targets that could help be preventative, so we never go down this path, is really important. Targets that we can go after to get to remission. Some people already have the disease or progressively down there. We're trying to get rid of flares, trying to get rid of, advancing the disease is really, really important. And certainly you get to that point where there's significant damage that goes on from the chronic conditions, and trying to prevent that type of tissue damage and identify that. So it isn't always like finding a new target or a new magic drug, but we want to do the full spectrum of what the patient experience is with these diseases and use this type of information we're pulling in to help in all those different areas, as well as the new drug development that's going on. Next slide. So we're getting started. This is just really initiation right where we're at, and we're doing a lot of early piloting. Now we got to finalize some of these standard operating procedures and get everything started. And then we're doing some pilot experiments. And even on a small number of patients, we're really able to get a wealth of data that can help us decide what's the right way to go after, what seems to be palpable that we can, do more, power additional analyses to get us started. And we're doing this through the different techniques that I showed you, a little bit more of the deconstruction and also that 3D reconstruction that we're doing. And it's going to allow us to really address these key questions that are there in the autoimmune space that you can see in the right, that I'm sure your doctors and yourselves, as you do your own interrogation of the work that's out there. But this is what we want to get to. This is what we want to advance to the field And it's great to have a dream team that's put together, but we also need the advocacy partners. We need to communicate what this means to you, how this is advancing the field, and they are a close and allied partner with what we're doing. I think that might be it.
Thank you so much, Steve. What an incredible presentation. I am so excited about what you're working on, and I cannot wait to see what happens next. So thank you for joining us, and I'm excited to bring up our next panelist, Dr. Arun Das. Dr. Arun Das is at Cabaletta Bio. He is the Chief Business Officer. They are working in targeted cell therapy, and their goal is to deliver deep, durable, and potentially curative, yes, curative responses for autoimmune patients. Wait until y'all hear about the science. You're going to be so jazzed. All right, Dr. Arun Das, you're up. Can you walk us through, tell us about what you're working on at Cabaletta, and then let's jump into some Q&A.
Absolutely. So thank you so much, Lily, for hosting us. Thank you to the Autoimmune Association. We're really thrilled to be here. It's an honor and a privilege. Wonderful. We've got the slides up. My name's Arun. I'm Chief Business Officer at Cabaletta Bio, like Lily said. Really thrilled to be here. Let me just tell you a little bit about who Cabaletta Bio is and what we do. We're a company that is based in Philadelphia, originally spun out of the University of Pennsylvania with some incredibly exciting scientific work by our scientific founders and the individual who is currently our CEO now. And when we were founded 5 years ago, we were founded with the following vision: to develop and launch the first curative targeted cellular therapy for patients with autoimmune diseases. So I really loved listening to Steve's presentation because he talked about personalized medicine. And what we do at Cabaletta Bio is personalized medicine aimed and designed to deliver potential cures for patients. We take patients' own cells, re-engineer them outside the body to recognize a specific signal on a specific type of immune cell. And we'll go through that a little bit more in the couple of slides to come, and then re-infuse those same patients, the patient's T cells, into their own body to deliver what has the potential to be a living cure, because those cells can recognize the disease-causing cells in the body, recognize, bind, and eliminate them. And the goal being to deliver lasting responses or lasting remission for patients with a broad range of autoimmune diseases. So this is what gets us up every day. This is why, we're 60 people strong. And what we try to do every single day is just make a little bit more progress to deliver on this vision. And so what are the approaches we're using to try to do this? And there are really two approaches. So the first, and I know this is a busy slide, so I'll ask for everyone's attention to go to the left side first. One of the approaches we're taking is called the CARTA approach, or chimeric antigen receptor T cells for autoimmunity. That's just a fancy way of saying it's a patient's own T cells that are being engineered to recognize a specific population of cells. And the way these cells are engineered is you have your immune system is made up of T cells and B cells. T cells are really the cells that are responsible for recognizing and destroying things that need to be destroyed. And so what we do at Cabaletta is take a patient's cells, isolate the T cells, which are the ones that are responsible for doing the killing, and engineer them so that they express a certain signal. And that signal is what you see on the left-hand side, the anti-CD19 targeting domain. That targeting domain targets a population of immune cells in patients with autoimmune disease that are actually the cause of the autoimmune disease in patients. So CD19 is a signal that's expressed on B cells in the immune system. And those B cells are responsible for producing the autoantibodies that are often the cause of the disease in autoimmunity. And so we aim to recognize those B cells, eliminate them. And what we've seen in some recent data that's been published actually last month in Nature Medicine was that a handful of patients with lupus that was moderate to severe and unresponsive to any medications that were provided to those patients were treated with this exact type of therapy, CD19 CAR T cells, and they were able to experience remission up to 17 months of disease in the absence of any other medications. They had a repopulation of their native immune system. And even with that repopulation of the B cells, no disease had recurred in patients up through the follow-up period. And so really why we're excited about that is because we believe this has the potential to reset the immune system in patients with a broad range of autoimmune diseases through a temporary B cell depletion and then eventual repopulation of those healthy B cells. So that's the first approach we're taking. There's another approach we're taking called the CART approach, or the AA CAR T approach, which you'll see on the right-hand side of the slide. There, what we know of is in certain rare autoimmune diseases such as pemphigus vulgaris or musk myasthenia gravis, we can actually identify a very, very narrow set of B cells that are responsible for driving the disease and producing the specific autoantibody that we believe is causing disease. And there, we take the same process. We extract patients' T cells, engineer them to express a certain signal, and then reinfuse them back into the patient. The difference with the CART approach is that the target that we have on the outside of the cell doesn't address all B cells. It only is designed to bind and eliminate the specific cells that we know of that are causing disease. Now, again, this approach is limited by the fact that many autoimmune diseases have more than one simple cause of the disease or one potential driver of the disease. But when we can identify that and engineer a cell to recognize those specific cells, that's the approach we're trialing. And so we're really excited about the trials that are ongoing and the trials that are anticipated to be up and running over the next 6 to 12 months, really being able to deliver on the promise of potential lasting remission for patients with a broad range of autoimmune diseases. And so, I've often been asked, well, how does this therapy actually work? What is CAR T therapy and how does it actually look when it's being administered to patients? And so this slide here is meant to give an overview of what the process is like to deliver this personalized therapy to patients. And so we have the leukapheresis or apheresis procedure, where cells are extracted from patients, collected from the body, and then sent off to a center where they are modified or engineered in the laboratory so that we can then expand those number of T cells that you see in the body. And that investigational product is then infused back into the patient's body in order to either recognize the CD19 CARTA approach, so all B cells that are expressed in a patient's body, or in the case where CART is feasible, the specific population of B cells that we believe are causing disease. And we have a clinical trial that's currently up and running in pemphigus for the CART approach. We have a trial in musk myasthenia gravis that's slated to initiate this year. And for the CARTA approach, the CD19 CAR T approach, we are aiming to file our investigational new drug application in the first half of next year and initiate the study thereafter in order to really deliver on the promise. So we're excited about this work. I really thank Lily and the Autoimmune Association for inviting us and would love to answer a few questions if we have the time.
Thanks, Arun. I'm just so excited about what you're working on. It's so promising and exciting for our community. And so, yes, I'd love to dive into a few questions. And my first is that, and you touched on this, but I'd love to know how this treatment is different from some of those existing treatments that are out there, like steroids and biologics, DMARDs, etc.
Yeah, it's a great question, Lily. Thanks so much. I think there are a lot of therapies out there, and I think each one has provided progress for patients, and we're thankful for any treatment that can do that. I think the biggest difference between the approach we're taking and what's currently out there is probably twofold. One is whether it's biologics, DMARDs, steroids, these are all chronic medications that exist in order to provide symptomatic relief for patients over a certain period of time. So whether it's a biologic, steroids, et cetera, and you can take these at different, periods. So some may be every day, some may be every 8 weeks, some may be every 6 months. But you need to take these medications, chronically in conjunction with other medications in order to see improvement of disease. The vision for the Cabaletta's approach is a one-time infusion that can allow lasting remission off all other medications. And so I think we always hesitate to use the word cure without seeing that it's actually the case. But that's the vision. That's why we were formed. That's what we want to do. That's one big one big difference, I would say. The second difference, and I kind of touched on it before, is so rather than being chronic, it's one time, and rather than delivering incremental improvement, we aspire for this to be complete disease remission. And so that's, again, what we're looking for in clinical trials, what we're excited to hopefully see, and again, why we're here as a company.
Yeah, and what a great segue into my next question. I just have to say, I think about the idea of not having to, stab myself with an injection every, whether it be every week or 2 weeks or a couple of months, or being remembering to take a pill. Like, it's just that's such an exciting concept, and I'm sure that a lot of our listeners can appreciate that as well. So you mentioned trials. What studies do you have ongoing, and how can folks learn more?
That's a great question. So we currently have a phase 1 study that's in patients with the disease. And so that disease is mucosal pemphigus vulgaris. It's a subset of pemphigus vulgaris that's an autoimmune blistering disease. That trial is up and running. We have a study from musk myasthenia gravis that's slated to start this year. So we're excited about that. And then for the CARTA approach, like I mentioned, there's not an active study ongoing, but next year we anticipate starting a study, more information to come on all of these. You can either find the active clinical trial listing on clinicaltrials.gov by searching for Cabaletta Bio, or you can go to our website, www.cabalettabio.com, and go to the For Patients page to find out more about information about the disease and the ongoing studies.
Fantastic. That's great. Thanks, Arun. And I encourage folks, if you missed our clinical trials session yesterday, to go ahead and watch that, because that's a great overview of what to expect with trials. And then, pop over to Cabaletta Bio's site and see if there could be something for you. I also love that you're starting in some of those rarer autoimmune diseases. Sometimes I know folks can feel like they're being left behind in the rare space, and it's important that we're working across all autoimmune diseases, the common and rare. And so I'm really encouraged to hear that that's where your focus is initially.
Thanks so much, Lily.
And finally, I'd love to end with asking what your personal hope is for the future of autoimmune treatment.
Yeah, it's an incredible question, and I appreciate it. When I came to Cabaletta Bio about 3.5 years ago, right near when the company was founded, it was with the following aspiration. As a clinician in medical school, all throughout my career, I had seen patients whose symptoms were trying to be controlled with the variety of medications that were out there. And the reason I joined Cabaletta was because I felt like this was the first opportunity we had to really completely transform the paradigm of treatment. And it went from managing symptoms to delivering cures. And that's what we hope to do. And we know it takes time. We know it takes a lot of effort, the engagement of patients, caregivers, stakeholders, physicians, other providers, just the whole network of folks to even get us where we are today, and hopefully to continue executing on that initial vision and aspiration is why we're here today. And we hope to be able to execute on that in the following months and years. And thank you for the support, and thank everyone for their engagement and support as we try to deliver on that.
Arun, what a thoughtful answer, and very encouraging answer. This has been amazing. Arun and Steve, I can't thank you both enough for contributing to this conversation, to sharing and giving hope. What this has done, I know for me, it has instilled a lot of hope in the future of autoimmune treatment, and I hope it has done that for our audience as well. I thank you for joining us. I thank you for fighting for our community, and I look forward to watching all of the amazing work that you're going to do. Thanks, everyone, for joining.
Thank you, Lily. Thank you all.